The next aspect of the chemistry of cells that we need to review is the importance of semipermeable membranes. And we'll be covering membranes extensively later, so this is just a little introduction. In all cells, the cell membrane is important for separating the cell from its environment. So in order for the membrane to do this, the membrane has to be semipermeable. It has to allow certain things in while keeping other things out. It also has to be insoluble in water so that it doesn't fall apart. The membrane also needs proteins that fulfill specific purposes like transporting material, responding to signals, and also attaching cells to other cells to form tissues. So the membrane is not just the barrier that separates the cell from its surrounding, it's also a site of biological activity. The cellular membrane is composed mostly of phospholipids, but it also contains glycolipids, which are lipids that have sugars attached to them, proteins, glycoproteins, and sterols, with the exception being bacteria, which do not have sterols in their membrane. In animals, the sterol present at the cell membrane is cholesterol. Phospholipids are amphipathic. They have a portion, the fatty acid tails, that is hydrophobic, and another portion, the polar head group, that is hydrophilic. So they have a phosphate and another polar group attached to the phosphate. Because of their amphipathic nature and as a result of the hydrophobic effect we discussed earlier, phospholipids spontaneously assemble into a bilayer when they are placed in an aqueous environment. So hydrophobic interactions occur between the fatty acid tails, while the polar head groups interact with water molecules outside the cell and inside of the cell. Since the interior of the membrane is hydrophobic, this means that there are certain molecules that are either unable to pass through the membrane altogether, or their movement through the membrane is greatly reduced. In general, a pure phospholipid bilayer is permeable to nonpolar molecules like oxygen, carbon dioxide, and steroid hormones. It is impermeable to most polar molecules, though some, like water, that are very small, are able to pass through, and it is highly impermeable to ions. Because many of the molecules that are present within cells are polar, like glucose, ATP, amino acids, their movement directly through the phospholipid is impeded by the hydrophobic environment in the interior of the bilayer. However, because water is a very small molecule, it is able to pass through the membrane directly by osmosis. As we'll learn later, besides osmosis, there is also another way that water can enter some cells. In the case of ions, although these are small, membranes are highly impermeable to them because they are charged. Recall that in the presence of water, a shell of hydration forms around ions, and that further impedes the movement of ions through the bilayer. The reason for this is that in order for an ion to move through this hydrophobic environment, that sphere of hydration would first have to be stripped from the ion. Because a pure phospholipid bilayer is impermeable to many of the solutes that are present in cells, transport proteins, which include channels and also carriers, allow these solutes to move into and out of the cells. We'll discuss the differences between channels and carriers when we cover membrane transport, but for now, it's important to remember that transport proteins play a key role in the selective permeability of membranes, because without these proteins, many of the molecules that need to move into and out of the cell would not be able to do so at rates that are fast enough to support life.